Okay, so we're going to be doing the last lab today. So this is on evolution, and as promised, it is a lot of math. We've been continuing on with this trend with the math. I'm going to do the best I can here to explain it to you. Hopefully you guys have been watching some of the videos I already put up on Hardy-Weinberg and stuff like that. So what we're going to do here is, again, you need to know your vocab. The second thing is there's kind of this understanding of evolution, but we can actually divide evolution into two kind of fields or ideas, and one is microevolution, is macroevolution. So most of us, when we think of evolution, we think of, for example, the progression of, you could say, dinosaurs to birds, or apes to humans, okay? And that would be macroevolution. And that would be the process of speciation, going from one species to another species. A species being defined as a group of individuals, which is a population, that interbreed and produce offspring that are viable. That means they live and are fertile, because it doesn't do you any good if they cannot reproduce themselves, like mules can't. Mules are not their own species. We get a mule if I'm crossing a horse and a donkey. The mule is sterile, so we cannot say mules are a separate species. They're actually a hybrid, a sterile hybrid of those two species. Okay, microevolution talks about things like small changes within a population. You can look at the microevolution of human height and how it has altered in getting taller in times where nutritional I don't say values, but nutrition has been more readily available. And so we maximize our potential for height versus times where food was scarce or the quality of food was not good. So we can look at the average height of humans and how it's changed over the generations. And that's kind of what we're focusing on in this lab. And this is called microevolution, and this has to do with gene frequency. So how often is that tall gene in the population now versus 100 years ago? So that is what we're looking for. We're looking at how often is that gene in that population. Is it fixed? Do we always have the same percentage, or does it wax and wane? Does it get higher? Does it get lower? All right. So in this lab, we are going to focus on microevolution. Okay, so you need to keep straight your alleles and your gene, which is your gene frequencies, and the difference between alleles and genotypes. So remember, an allele is represented by a letter. So in our lab, we're going to use big A for the dominant allele, and we're going to use, and I hate autocorrect. That should be a little a. So little a is the recessive allele. So do make sure that you have changed this, and I can annotate. Of course, it's just going to take longer, but that should be a little a. Okay, not a, big A. Get rid of that. Ugh. All right, little a is the recessive. So we have here, when we talk about a genotype, now remember, genotype means you have two alleles, one from mom, one from dad. So if you are homozygous dominant, you have big A, big A. If you're heterozygous, you have big A, little a. If you have, and there's my autocorrect again. If you have little a, okay, little a, you're homozygous recessive. All right, so do make sure you make note of those corrections there. All right, so again, we've been using these genotypes and this vocab now for almost 3 labs. So we need to have our genetics down. Okay, so now I have to, let's see, I have to erase everything before I go to the next slide, or it'll be there forever. All right, and then close that. And now I have the power to advance. Okay. Ooh, don't want that much power. All right, frequency. So when we ask about frequency, we're asking how often that allele or genotype appears in the population. So if I have a population of 330 individuals, I'm going to ask you, well, how many alleles do we have then in that population? Well, remember, we have 2 alleles per person because they have 1 copy from their mom and 1 copy from dad. So that means we would have 660 alleles in that population. So again, if you don't remember 330, they each have 2. That gives me 330 times 2, 660. That's how I got that number. You'll have to get that number too on these labs. So when you set up your gene pool, what you're going to do is it's going to ask you, so if you have your lab book with and you're following along, you should be on page 166. And what you're doing is assembling a stock population. So you have these 3 containers, one that says your gene pool, one that says live, one that says dead. And we have all these red and yellow beads, and you're asked to establish your original breeding population. And so they're saying in that original population, you have 25 homozygous dominant, 50 heterozygous, and 25 homozygous recessive. So in order to figure that out, so again, our A is the dominant allele in that little a. That's gonna vex me through this whole thing, I'm sure. And it doesn't matter how many times you cross it out and you keep going back in and say little a, it just keeps putting it back in. I got it. All right. So it's asking you how many beads you got to start off with. Now, if I go with my dominant allele, so my dominant allele is going to be, I have 25. That means I have 2 dominant alleles. So my 25 in each individual has, and I'm actually change the color if I can figure out how to do that. Text, give me my color. Apparently I can't figure out how to change color, so I am stuck with blue. It's a good thing I like blue, but it doesn't show up too well. All right, so we're gonna stick with that for time's sake. 25 times 2, that gives me 50, okay? But now that gives me 50 with my homozygous dominant. Now notice I also have, let's see what this does. I also have oh dear Lord, I don't know what's going on with my computer because it just asked me if I wanted to get out of Zoom. So hopefully I'm still recording. So I have, well, I was going to try to highlight, but apparently it's not going to do that. So I have, remember I have, not only do I have the 25 homozygous, but I also have 50 heterozygous. So I also have an additional 50 big A's. So I have to add that together. So my 50 plus my 50 is going to give me 100 red beads. All right. So I have my original 50, and then I have my my 50 from my heterozygous. Okay, so now what's going to happen, if I don't throw my computer out the window, is I have to figure out my little a. So my little a is going to equal, well, I have 25 times 2, which is going to give me another 50 yellow beads. But that's not all the yellow beads I have, because I also have that 50 heterozygous. So that's, I have to add 50 more yellow beads. So that gives me also a total of 100 yellow beads. Okay, so I have 100 red beads and I have 100 yellow beads, and you guys would have counted those all in class. Okay, so you would add 100 and 100. And so again, we would need 100 red beads and 100 yellow beads, and you would have put them together in your live or your breeder cup. All right, so that's all you're doing with the first lab, is just establishing your population. And so then you would have then gone to answering the next few questions, and that is filling out the table. And when we look at filling out the table, what we're going to do when we fill out that table is that we are also going to, I'm going to try to do this with you as well with the little insert button here. So now we know our number. So we're dealing with frequencies. So we know that we have a total number of, right? So I have, how many do I have here? I have 100. I have 100 genotypes, right? I have 25 homozygous, 25 homozygous recessive, and 50 heterozygous. So that gives me 100 individuals, right? These are individual people. Those are their genotypes. So to get my frequency, what I need to do is I need to take my 25 divided by 100, and that's going to give me 0.25. And then I'm going to do the same thing for my 50. I'm going to go 50 divided by 100. That's going to give me 0.5. And then I'm going to do my last column, which should be the same as the first one, right? 25, 25 divided by 100, which is 0.25. Now notice this adds up to my 1 to 1 or 100%. That better, because if it doesn't, you're missing some individuals in your count. All right, so now that I have this, now I need to figure out my gene frequency. So with my gene frequency, I want to know, well, I had 100 red beads out of 200. So that's going to be 100. So I had 100 red beads out of 200 total beads. So that's going to give me 0.5. I wish I could just cut and paste that, because you're going to get the same number for this one, right? Same thing. It's 100 divided by 200. Now it's going to give me 0.5. Which is right. My frequency should, again, my dominant and my recessive allele should add up to 100%, or again, I am missing somebody. All right, so you need to be very careful of that to make sure your numbers come out. So again, that should come out to 1 or 100%. Now, if we wanted to go backwards when we do this in the future, what you're going to do is you needed to count your how many A's. So if you look at that again, you would have 25 times 2, which is 50, plus 50 here, which would give you 100. 25 times 2 here would give you 50 little a's, plus 50 here, which is 100. All right, so that is what we did. So get rid of this. Now, the next slide, what we're going to do is we're just going to answer some questions. So here's all filled in for you. Notice there, 1, 1. So what does the red bead represent? Okay, so when we talk about what does it represent? Well, it's representing the dominant allele. And then what is the probability that you're going to draw out a red bead? So again, when you draw in, it's a 50-50 chance, right? Because you have half red, half yellow. So it's 50% chance. And then what's the probability of drawing a yellow bead and then another yellow bead? So this is back to our and or rule, and we're asking for yellow and a yellow. So what is the chance of getting a yellow? Well, the chance is getting 1/2. What's the chance of drawing out a second yellow? Well, 1/2. Well, we want both of those to occur, right? We want yellow and yellow. So we got to multiply those. So when we multiply our 1/2 times 1/2, we get 1/4. So we have a 1 in 4 or 0.25 chance of getting a homozygous recessive genotype. Now, with our Hardy-Weinberg equilibrium, these are two scientists that discovered that populations will not evolve if the following conditions have been met. Now, if we look at all 5 of these conditions, we can honestly say that the odds of that happening is slim to none. Therefore, they're basically saying all populations are evolving. So the first one is, well, there has to be no mutation. We get mutation. That's a common thing to get mutation. We're constantly making mistakes when we replicate our DNA. Sometimes it gets fixed, sometimes it doesn't, sometimes it gets through, sometimes it causes a problem, sometimes it doesn't. No gene flow. So what we're saying here is that when we talk about gene flow, we're talking about populations intermingling with other populations. So when you guys would sit in a class, each of you would have your own separate populations, and that's what something I would say is make sure you have no gene flow, because you don't want your beads getting mixed with another group's beads. Then you're migrating alleles, okay? So no gene flow. Keep your population to itself. We know that doesn't happen. Populations migrate all the time. Random mating. It has to be completely random. It can't be that, this female's picking this male because he has these positive characteristics. Nope, it's got to be completely random. Just like you reaching into your breeding population and pulling out two beads. You have no idea which one's red or which one's yellow. And a large population size. So it's got to be fairly big. Of course, I don't have a specific as to what is the size that is large. So that's kind of a discretionary size. It's not very quantitative. Is it large? What's large? All right. No selection. So again, we cannot be picking for certain characteristics. Okay. So that's kind of what we're trying to demonstrate with the beads. The beads are the same size, the same shape. The only thing that's different is the color. And if you're blindly drawing two beads, you have no idea whether they're red or yellow. So this is where we're hoping to show you that we would get no evolution occurring, at least microevolution here. That's what we're talking about. Mind your P's and Q's. Okay, so in this lab, it is very important to mind your P's and Q's. So what do we mean by P's and Q's? So first of all, in a population, our alleles. So our P's are our dominant allele. Our Q is our recessive. So we know that if we count up all of our dominant alleles and all of our recessive alleles, you're going to end up with 100%. That's 1, right? So whatever that percentage is of P, what the other half, so if it's 80% P, the Q's have to be 20% because it has to add up to 100%. Now, we also know this formula, which you learned in math class. And if it starts bringing you back memories of the quadratic formula and FOIL and all that crap, teacher did a good job because it's scary stuff. All right, we're not going to be getting into that scary stuff. We just need to know what these terms mean. Okay, so what do these terms, P squared plus 2PQ plus Q squared, is going to total up your whole population? So what we're saying here is our homozygous dominant plus our heterozygous plus our homozygous recessive will equal 1. Well, you shouldn't have any other gene types because you only have big A, big A, big A, little a, or little a, or something's really going on weird. Okay, so with that information, if you remember what is what, this is probably one of the the most important slide in this PowerPoint. When you do your word problems, you need to remember that P is the frequency of the dominant allele. If we have two P's, so that's like basically saying P could be the same as big A. Well, that's an allele. If we want a genotype, that's going to be big A, which is the same as P squared, right? P times P squared. Q is the frequency of the recessive allele. So therefore, again, Q times Q is going to be your homozygous recessive, which is little a. So Q squared is little a. 2PQ is your heterozygous. I'm saying I have a P and a Q, which is big A, little a, but I can also have little a, big A. There's 2 ways to get a heterozygous: big A, little a, big A. All right, and so that's why you have that number 2 there. You have 2 ways of getting the heterozygous. So that formula is 2PQ. All right, so if you look at procedure 12.2, what you do is you have this population. So we should be on page 170. You have this population, your 200 beads in there, right? 100 yellow, 100 red. And what I have you do is I have you reach in and pull out 2 and record the genotypes. Is it yellow, yellow, red, red, yellow, red? And then you put it in a different container and you pull the next 2. And you keep doing this, okay? So actually what you're gonna do is you keep putting them back in the container, okay? So you pull on your cord, put it back, and you do this for 100 pulls. So 100 times you do this, and then you record it. So you and your partner would have recorded it, and in this case, you recorded 25 red, red, 48 red, yellow, and 27 little yellow, yellow, right? That's little a, little a. Or 25 PP, 48 PQ, 27 QQ, right? So then I just took that and divided them by 100 to get my frequency, 0.25, 0.48, 0.27. And then I went over here and I had to calculate my A and my big A, little a. Now, again, to get that big A, little a, what I had to do was I had to, because this is not, we have our initial population. So now we're like, well, what did we get when we did our pool? And it's going to be really close to our initial population because we really did not exceed, we met all of the requirements for Hardy-Weinberg. And so what we're going to do is for big A, that's going to equal, remember we had 25, but we have 2 of them, right? Big A, that's 2 big A's. And then we also have the heterozygous has 1, and we have 48 of those. So we have to add those up. And so when we do that, that gives me 98. And then I'm going to take my 98 divided by 200. And that's what gives me my 0.49 for my frequency. Now for my little a, well, I have 27 homozygous recessive, so that's 27 plus 27, right? They have 2 copies. And then my heterozygous has 1 copy. So I have to add that one in there, my 48. All right, so that gives me 102, and then I divide that by 200 because that's my total, and that gives me 0.51. That is really, really close. Okay, well, how close is it, and is it significant? Is there a significant deviation? Well, we know a test we can use to test that, and that is your chi-square test. So what you're going to do is you're going to take those genotype numbers 25, 48, 27 and put those into your observed column. Now, we are going to assume, based on our original population, that we have a 1-to-1 ratio. Okay, so with our 1-to-1 ratio, you should be able to figure out your expected and get your chi-square test. So what I would like you to do here is, in order to practice your chi-square and make sure you know how to get your expected, I would love it if you actually did take the time to pause this video here and then fill out your chart and then check it with the answer that I'm going to throw up here in a second. All right, so go ahead, pause. All right, so there is my math. Hopefully it's fairly close to yours. Of course, there may be some rounding differences, but I got 0.24. All right, so hopefully that's really close to yours. So what you're gonna do next is you're gonna say, well, I know right now, well, I have 3 outcomes, so my degrees of freedom is 2. And if you actually look at the bottom of the table, on the top table on page 170, they have a little breakdown of the chi-square chart for you, and it says that your allowable chi-square value is 5.991 with your degrees of freedom being 2. So what does that mean? That means I am way under what my allowable. So when I look at my questions, so it asked, did my population evolve? Well, no, it did not. It is less. My 0.24 is less than 5.991. Okay. Oh, I don't know why my other, oh, I know why. All right. So the next is going to start with some word problems. Now, with these word problems, what we are going to do is we're going to be using a lot of the terminology that I have been talking about. So with that word problem, okay, let's go through and I'll work these 2 problems with you. So it says the frequency of your dominant allele is 90% in the population. So what does that mean? That means P equals 0.9, right? That's the same as 90%. What's the frequency of the recessive allele? So what is Q? Well, remember Q, I can figure this out because P plus Q equals 1. Therefore, my Q has to be 0.1. So now I know my Q. Okay. Now, the next thing it asks is what is the frequency? Well, I did that one. What's the frequency of the recessive allele? So that's my Q right there. What are the frequencies of the homozygous dominant? So if I'm asking my homozygous dominant, that's big A, big A. Now remember, that's also the same as P squared. I don't know where to do that little up button, but that's the best I can do. So I'm going to go 0.9. Let's try 0, not a O. Times 0.9, and that is going to give me probably 0.81, which is the same as 81%. And then it asks you for the heterozygous. Remember the formula for that. So that would be big A, little a, and that would be my 2PQ. All right. So 2 times my P, 0.9, times 0.1. And I type that in my fancy schmancy calculator, and I will get 0.18 or 18%. And then it asks you for the homozygous recessive. So my homozygous recessive would be little a, or which is the same as Q squared. So that's going to be 0.1 times 0.1, and that is going to equal 1%. Okay, so or 0.01, which is, I did this right in my head, 1%. And if I notice, my 81 plus 18 plus 1 gives me 100%. Everybody's been accounted for. Okay, so that is how I would figure out this problem. You'll have a lot of these problems to do in the review. I highly suggest you go through them and you check them. I would check them way before our review on Monday, so that if you want to sit in my review or you need to Zoom with me, please do, because this is, I mean, there'll be at least probably 4 questions like this. So you need to be able to do this. All right, problem 3. 25% of a population, so we're saying 25% of a population is homozygous dominant for a particular gene. Okay, that's my genotype, which is also my, and they're saying it's dominant, so that would also be my P squared. So how do I get P? So in order to do this, and I do not know how to draw this, we don't have symbols in this, is you're going to have to take the square root. So you're going to have to take your 0.25, and then you're going to have to take the square root of it. So, because right, 0.25, and then the square root. And that is going to give me, because right, P squared, in order to get rid of the square, I gotta take the square root of it. So that is going to end up giving me a P of, so my P is going to equal 0.5. Boom, now I know my Q, right? My Q is going to be, right, because P plus Q is 1, so my Q, so that's going to equal also 0.5, because 0.5 plus 0.5 gives me 100%. Okay. So what are the frequency of the dominant recessive alleles? Got them, 0.5, 0.5, and now it's going to ask me what's the frequency of the heterozygous. That's my 2PQ. So 2 times 0.5 times 0.5, that is going to equal 50%. And then it's going to ask me for my homozygous recessive. What is my Q squared? Q squared. And so again, that's going to be 0.5 times 0.5. And that's going to give me 25%. And that should come out right? 25% plus 25% plus 50%. That gives me 100%. My population is all accounted for. So again, you need to make sure you realize what's a genotype, what's an allele, what's a genotypic frequency, what's an allelic frequency. Okay. And so then they start going into natural selection. Right. So hopefully everybody's good with the math. I'm gonna erase it all here. All right. So moving on to the next lab, which is natural selection. So with natural selection, what we're going to do here is that you're going to pull 2 beads out at a time. And every time you pull out 2 yellow beads to the homozygous recessive, you're going to throw them in the dye container. They're not good. They get picked off. Predators grab them. All right. So pick out your beads, throw them in there. Pick out your beads, throw them in there. The ones that are either red, red, or red, yellow, live. Yellow, yellow, dead. Red, red, live. Yellow, red, live. Yellow, yellow, dead. You're going to keep doing this until your container's empty. And then you're going to take all your yellows and put them aside because they're not coming back. They're dead. And you're going to take your new established population. And then what you're going to do is you're going to reach in and you're going to pull out until your thing is empty, which is why everybody gets different numbers because it just depends on how many beads you got rid of. All right, so you're going to pull them out until it's empty and record what your genotypes are, whether they're red, 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 yellow, or yellow, yellow. Okay, so this is going to be what your numbers, so this is what you got. All right, so write down your numbers. So you got 20 homozygous dominant, 37 heterozygous, and 10 homozygous recessive. So you got 60 individuals. You then need to figure out your frequency. So then you're going to go your 20 plus 20 plus 37, and that's going to give you 77. And then you're going to divide it by, remember, you got to go 67 times 2. So you're going to divide that number out. I think it's 134 to get your frequency. So hopefully you see now we've got a frequency change in this population. And we're going to do the same for little a. Little a, that's 20, right? 10 plus 10 plus 37, that gives me 57, and then divide that again by 134 to give me my frequency. So again, the frequency of alleles would be how many letters, and we got to multiply 67. We have 67 individuals, but each individual has 2 alleles. So that's where I got my 134. I took my 20 divided by my total number, 67, to get my frequency of my genotypes. Next, you're going to do a chi-square analysis to figure out, well, is this significant? Is this a significant change? Do we have evolution? Do we have outside our number that we would expect for just a probability of not being exact? So again, I would suggest you pausing the video and doing your chi-square, making sure you have that right. Again, we have a 1-to-1 ratio as our expected because we started off with 50-50 with heterozygous population. And then I would restart the video and check your answers. All right. All right, so if you're back with me, here we go. So I got 3.72, which is still pretty small, seeing as that our allowable chi-square value is 5.991. So when I answered my question, did my population evolve? Well, we're still within an acceptable deviation. So no, my population did not evolve. So even though we did have some natural selection here, there was not enough. And again, it really depends on your pool. I have some students that will get they'll get evolution and some that don't. It's totally chance on how they drew out, how many of those yellows they got rid of based on their polls. Okay, so then I would answer those questions on 173 and then check them I would check them in the module. All right, so again, check your answers with the key. Then the last procedure that we're gonna do has to do with what's called the bottleneck. Now the bottleneck talks about like if a population has this situation where there is this catastrophe and many individuals are just killed out of the population. And it's completely random. It doesn't have to do with their characteristics. Any traits that they're carrying, it is completely random. Like if a volcano went up, it wouldn't matter if you're tall or short, dark skin, light skin, smart, stupid, wouldn't matter, you're dead. Okay, so this is what we talk about with the bottleneck. And then it's like, well, who survived? Well, in this case, you only had 4 individuals survive. So you're going to randomly pull 8 beads out of your population and put them in the live cup, and that's what you're going to pull from. And so you're going to reach in, pull 2 beads, record, put it back, reach in, 2 more beads, record it, put it back for 40 pulls. So you're going to do 40 counts and recordings. So this would be your number. So I said when you did it, you got 10 homozygous dominant, 15 heterozygous, and 15 homozygous recessive. So there's your 40, and then I figure out my frequency. I took 10 divided by 40, 0.25, and then 15 divided by 40, 0.375, and then 15 again, 0.375. For my frequency, I had to go 10 plus 10, that's 20, plus 15, that's 35, and then divide that by 80. I have 80 alleles. I have 40 individuals, but each individual has 2 alleles. And then for my little a, I have 15 times 15, that's 30, plus another 15 here. So that gives me 45 divided by 80, which gives me 0.56. So we do have a little bit of deviation here. Now this lab is really dependent on your poll. I have students that I will walk around my room, and half the time I'll have some students will get, well, they happen to get 40 red and, or 4 red and 4 yellow. And so they don't really see an evolution. I'll have some students who will get like 6 red and 2 yellow, and it's definitely going to show that there was evolution. So this particular lab result really does depend on the class, on your group, and how many people survive, what alleles survived through that catastrophe. All right, so again, I have another chi-square. I would check your chi-square. Again, I'm going to have you guys pause it, do your calculations, and then check for your calculated chi-square. You will have several of these to do on the lab practical as well. So pause now. All right, if we're back, this is the number you should have gotten close to, 3.75. So when you look at that, did our population, no, we did not evolve. Like I said, if we were in a class, we would see that some did, some didn't. And that's why it does ask, do all groups in the class get the same results? And that would be no, they don't. It really does depend on what survived their catastrophe. Okay. And then it says, will the effect of genetic drift? So this is question 3. What effect will genetic drift typically it does cause evolution, but there's no guarantee. It is completely random, but it does ask, what does it typically cause? And a lot of times you lose a lot of genes alleles when you take out a large population. You reduce the variability, the genetic variability within that population. So typically it does cause evolution. All right, and so that is the last lab we would have done. I would be I can't stress this enough. I would be it's very important that you work on these math problems on the review. All right, so with those, what I'm going to do is I will probably, like I said, I will probably have, if you need me to answer questions, I will answer questions before they are due, because again, I'm going to be live on, let's see, on Monday So you'll have the answer key will be there. So you'll check your answers because I have that released. And then on Monday, especially if I know you're asynchronous, but you definitely may want to attend our review for the lab practical on Monday night, or try to get a hold of me so we can Zoom or whatnot if once you check your answers, you don't understand how I got those results. So really be careful with your math. All right. So, and again, reach out if you need help. All right, hopefully this is helpful, and good luck with your lab. And I hope you guys had a nice semester. Bye.